Thank you.
Great. Okay. As we're at a time, let's get started. Thank you for joining webinar with us today. My name is Junyoung Park, KCITY coordinator. Since the director, Professor Jeong Kim, is out of town, I'll be the one who's hosting on the behalf of KCITY. Those who are not familiar what KCITY is, we are the GITCC center located at Yale. I'll be the hosting on the behalf of the KCITY for sure, and we are one, yeah. And then to inform that our mission is that holding a webinar that has a great meaning to introduce research area of Yale to Korea, effectively seeking the joint R&D, which is very important step in according to enter the global market. It is my pleasure to introduce our speaker, Professor Claudia C. Professor C is an assistant professor of electrical engineering at Yale University, where she leads the brain body bioelectronics lab. Before she joined to Yale, she received her PhD from Columbia University with postdoctoral training at MIT. Her work focuses on developing soft, flexible bioelectronic devices that can interact with the nervous system and peripheral organs, if I'm right.
Mhm.
In other words, communication between the brain and the body, including the gastrointestinal system. Her work led to the development of fully flexible, implantable bioelectronic system made entirely of soft but comfortable materials. She has also been recognized as one of the MIT Technology Review 35 Innovator Under 35 program for her contribution, which also suggesting that she's one of the rising stars, I believe, for sure in the field of bioelectronics and neutral interface technologies. By the way, Professor Sia, I also study in the UC San Diego too, so we have a lot of common things. All,
Nice.
although she is going to present all the details, Professor C will present her work unconformable bioelectronics, developing soft, wireless, and barrier-free implantable devices for interfacing with the brain and enabling the next generation bioelectronic therapies. The presentation is about, right, 30 to 40 minutes long, with the
Mm,
following the
yeah.
Q&A session. And with that, it is my great pleasure to invite Professor Clara C. to give her talk. Professor C, it is on your floor. Please. Yeah.
Thank you so much. Hello, everyone. Thank you for having me here. It's a great pleasure for me. I'm Claudia Shea, and I'm an assistant professor in the electrical engineering department here at Yale, where I lead the brain body bioelectronics lab. And today I'll be talking about conformable bioelectronics. Before starting, a little bit about myself. As you can probably tell from my accent, I was born and raised in Italy, where I did my undergrad in biomedical engineering at the University of Pisa. For my master's degree, I moved to the States, specifically San Diego State University, for studying biomedical engineering. And that's where actually I got my first approach to research working on neural interfaces for neurotransmitter detection. For my PhD, I moved from the West Coast to the East Coast at Columbia University in the city of New York, where I got my PhD in electrical engineering, working on neural interfaces for electrophysiology. And after my degree, I worked for 2 years at MIT as a postdoc with Professor Polina Anikeva in the material science and engineering department, working on devices for gut electrophysiology. And on July 2025, I started as an assistant professor here at Yale, where with my group we work on multimodal bioelectronic devices for therapeutics. But enough about me. Let's dig into the presentation. I think we can all agree that there is a strong demand for personalized medicine due to significant variation in how individual responds to medical treatment, particularly for patients that are affected by neurological disorders. A solution for that is bioelectronic devices that can actually deliver this personalized treatment by monitoring the physiological activity, identify biomarkers for local and responsive delivery of intervention, and then transmit this recorded data outside of the body for evaluation of the treatment efficacy. Nowadays, there are many great examples of bioelectronic devices in the market. We have different types that are targeting different organs and different diseases for different applications, spanning from the UT array for electrophysiology, the Neuralink, different type of pacemakers from Medtronic, the NeuroPace that is used for treating epilepsy, and so on. However, there are some challenges related to these devices that prevent the implanted bioelectronic devices widespread adoption, especially in chronic settings due to potential tissue disruption. They basically don't allow us to have these bioelectronic devices hosted in our body for very long period of time. These limitations are mainly three. First of all, the mechanical mismatch with the front end of the device and the organ that can cause implant failure or can lead to an inflammatory response. So oftentimes these devices are rigid, but they have to interface with quite soft body tissue or quite soft internal organs. Secondly, most of these devices have bulky backend connections that are needed for both the powering of the device and also for the transmission of the data, of the recorded signal. So these bulky backends, so very often wires or rigid components, they minimize their usage, the usage of these devices as wearable and chronic electronics. As we can see in this picture here in the center, we say this patient have this quite bulky device is the NeuroPace that is implanted in the skull, so that might cause some discomfort to the person. And finally, rigid encapsulation to avoid contact with body fluids. Most of these devices are encased into rigid metallic cases to protect all the electronics that is inside from the body fluid. So all of these elements together create some limitation for the widespread adoption of these devices for long-term operation. So what would be ideal to have in the world of the bioelectronics is a device that is fully flexible and conformable, where basically all the bioelectronic device components, starting from the front end, which is the part of the device that is responsible for the acquisition of the data, the processing unit, which is responsible for amplification and signal processes, and the wireless communication that is needed for the transmission and powering would be entirely soft and conformable without any rigid component, with the final goal of creating a fully flexible bioelectronic device. So this is the main mission of my research group, and this has been my mission since I started doing research. So developing devices that are highly biocompatible and conformable that can be nicely hosted by the body without rejection, so they can stay for long term with characteristics that are very similar to our organs. So starting from the front end, which is the device part that again is in direct contact with the tissue and is responsible for the acquisition of the signal, throughout my research I developed highly flexible and conformable neural interfaces based on thin film plastics, gold for the interconnects, and biocompatible polymers for a total thickness of this device of around 3-4 micrometers, so thinner than a human hair. As we can see from this panel on the right, these devices can be microfabricated in different form factors, for example, as surface devices that can be laminated on different biotissue from the palm of the hand or from the brain cortex, but they can also be fabricated as intracortical devices. So these what is called shank devices that can be inserted inside the brain to have a more precise and high resolution recording of the neural activity. Um, One main characteristics of these devices, besides their high biocompatibility and conformability, is also the performance in acquiring the signal. As we can see actually from this previous picture on the left, we see that these electrodes are made of gold interconnects, and then we see these little black dots on top of it that are made of conjugated polymer. There is deposits on top of this electrode to improve the performance and to improve the quality of the signal that is recorded. This type of polymer is called PWSS, which is in a liquid form and then can be deposited on top of our probes. And as I mentioned earlier, the main reason is because it improves the performance of the electrodes by reducing the impedance. On top of that, it's also easy to process and requires low temperatures handling. In general, the microfabrication of these devices is quite cost effective and is cheaper compared to silicon-based microfabrication. All the materials that are used and involved here are not only highly biocompatible, but also FDA approved, which in US means that basically you can use the material in human bodies. But yeah, the main reason why we use this conjugated polymer and we place it on top of our electrodes is because, as we can see on this plot on the right, we can decrease the impedance of this electrode while still maintaining a small dimension of the electrode itself. That in the case of recording high resolution neuron activity, you need the electrodes to have the same size of the neuronal body and the same spacing. So we see here that compared to a bare metal electrode, the impedance of these PWSS coded electrodes decreases by increasing the thickness of the coating. So basically, we can improve the signal to noise ratio of the recorded signal and the overall signal recorded. So as a matter of fact, we use these highly flexible devices to record a different type of biosignal, covering the entire electrophysiological spectrum. We recorded ECG activity from the heart, EMG from muscles, local field potential from a group of neurons, and action potential from single neuron, which is the most precise and the highest resolution type of signal that you can record when it comes to neural activity. And all these signals were actually recorded with a signal to noise ratio that was higher compared to commercially available metal electrodes. More in detail, we see here how we use these very thin and highly flexible devices that we laminated on the surface of the skin, and we were able to record the muscle and heart activity from the from the arm of the patient. And more invasively, we also use this same type of devices for intracortical recording, so all the way inserted in the neural cortex, where we were able to record again the activity of a single neuron. But the main characteristic of these devices is that due to their high conformability, biocompatibility, and also improved performance due to the spin coating of this polymer on top of the electrodes, we can actually record the activity of a single neuron even from the surface of the brain, which is something that, as I shown in the previous slide, is achieved by having intracortical electrodes. These specific devices here that we see in this picture on the left were actually used for recording from the human cortex. The experiments were basically performed at the Baylor St. Luke Medical Center and at the Columbia University Irving Medical Center, where basically the patients were undergoing placement of the brain stimulation electrode to treat movement disorder, and our device was placed on the surface of the exposed cortex to record the neural activity. Moving forward to the processing unit, so I've shown so far how using this technology we can develop a highly conformable front end, which again is the part of the device that is in contact with the tissue responsible of the acquisition of the data. Moving towards the processing unit, which is the device part that amplifies and processes the recorded signal, what we thought in order to have an overall fully soft and conformable device is that if we focus on what is the most essential component of a bioelectronic or in general of an electronic device, if we make this building block, so if we make the most essential component to be inherently flexible and conformable, then we can create an electronic device or a bioelectronic device that is highly flexible and conformable. conformable. So specifically, we focused on the transistors. I mean, we have billions of transistors in our phone, in our computer. However, they're mostly based on silicon, which is even if it has like unmatched performance, it is rigid in nature. So we can see the difference between these two pictures on the left and on the right. How a chip that is made of rigid transistor, it's overall rigid, while if we make a chip of transistors that are inherently flexible and conformable, can actually create an overall structure that is flexible and conformable. So for this reason, in order to achieve that, we focused on this novel type of transistor that are called organic electrochemical transistors. I will go briefly over the concept of how this organic electrochemical transistor works. Similarly to the electrode that I showed in the previous slides, these transistors are made entirely of biocompatible materials. We have different subsets that can be flexible or stretchable. Then we have the gold interconnects. And then the main characteristic is the fact that the transistor channel material is made of the same conjugated polymer that we use for our electrode to improve the impedance. Even in this case, the most wide used polymer is P2PSS that again can be deposited on top of the transistor to create the transistor channel. And the main characteristics about this transistor is that they actually really like ions, which is something that is very important if we want to create circuits that have to interface with the body fluids. As I mentioned in one of the very first slides, one of the main limitations of currently commercially available bioelectronic devices that they need to be encased into rigid metallic boxes to protect the electronics from the ions. In this specific case, these transistors they do actually ion, and their performance depends on the presence of ions. This is because this polymer that we use to make the transistor channel is hydrophilic. So when it's in contact with a biological environment, ions can actually penetrate into the bulk of the material, so into the bulk of the transistor channel, and perform this ion to electron transduction with a local amplification. So this transistor can be used not only to create the building block of circuits, but also to actually perform a local recording of the activity with a local amplification. So I spent a lot of time trying to improve the performance of this transistor. Of course, there are new type of transistors, so the overall field is still working towards improving the performance. And specifically during my PhD, I developed this novel architecture for organic electrochemical transistor that allow me to improve the amplification and the speeds, and to give a number to quantify this improvement. We calculated the ratio between the amplification and the speed, which are the two of the most important characteristic of a transistor. And we see here from this plot on the with the star how the performance actually of this transistor surpassed any other type of organic transistor and compared favorably with the top performing inorganic flexible transistor. Another important thing when creating circuits that directly interface with the biotissue, it could be the brain or any other organ, it's also important that the dimension of this transistor can be scaled down so that we can pack a lot of this transistor in a few surface area. And as a matter of fact, I was able to develop this flexible, very large scale integrated circuits where basically we put together around 1 million transistors in a few centimeters squared area while still maintaining the high flexibility of the overall chip with a total thickness of, again, 3-4 micrometers, so thinner than a human hair. And as we can see from the picture up on the left, this chip were like, we laminated this chip on the finger of the alma mater at Columbia University. But more importantly, we use these transistors to build some circuits, some flexible circuit that could be microfabricated directly on chip on the flexible device, such as amplifier, oscillators, and multiplexer. Another important element is that, of course, this transistor would operate stably over time in a physiological environment. So I show here how the performance of the transistor, the transconductance specifically, stayed constant for a period of time of around 1 year. And the only reason why we have these 62 days here in the plot is because I had to graduate, so I left the lab, but not because the transistor stopped working. So leveraging all of these characteristics of these transistors, we built the first prototype of a signal processing unit directly on chip, so direct on these highly flexible neural interfaces. Specifically, we developed a circuit that could detect real time, that could perform real time detection of epileptic activity in vivo. So we see here that the overall device was only this, again, very tiny shank-shaped intracortical device that was inserted in the hippocampus. Of the mice. This is the animal model that we use for this experiment. And we, as we can observe here, this circuit was able to detect only the epileptic activity. In fact, if we look closely at the black trace, these are just like the raw data that were recorded by a simple electrodes, while in red we have the activity that was recorded by this circuit that was built using this transistor. So we are basically able to identify only the epileptic activity and filter out the rest of the signal. So I've shown so far that with this technology we can build flexible front end, flexible processing unit. Now another important element, of course, of a bioelectronic device is the wireless communication, which is needed again for transmitting the data that we acquire from the implanted device, but also for powering it. And again, we want to still do this while having an overall device that is soft and conformable. So to do this, also for what regards the wireless communication, we use this technique that is called ionic communication that leverages the presence of ions that everyone naturally has in the body to transmit signal. Particularly, this ionic communication, just simply IC, permits flexible ion-based power and data exchange with the implant. The way it works is that basically if you have two pairs of electrodes that are separated by an ion-rich media, such as the skin, for example, you can transmit a signal from the transmitter. It will be seen the receiver through an ion-rich media by a change on the electric potential energy. And this depends, of course, from the quantity of ions that you have in the media. The more the ions, the stronger is the communication. But of course, if you imagine when you put an electrode into in contact with an electrolyte, there is the formation of the double layer capacitance. So in order for this system to establish a communication link, you need to overcome the double layer capacitance. So as you can see, this technique has this bandpass filter waveform where basically there is a specific frequency band where you can establish this communication link, and that again it changes and it shifts based on the quantity of ion that you have in the solution. And to give an idea of how flexible and conformable is this type of communication, the only thing you need to establish it is like two highly soft and conformable gold pads, again separated by an ion-rich media, how they're displayed here in this picture where we laminated the gold contacts onto a flower leaf. So we took all of these elements together and we developed the first wireless, battery-free, fully flexible device, that neuroelectronic device that was able of recording neural activity. So basically, we put together the ionic communication with our transistor and the flexible neural interface to have an overall neuroelectronic device that could perform electrophysiology, so recording at high resolution without the use of any rigid component. The way we did that is that we used three pairs of gold electrodes. Three, of course, were the implanted device, which was in the brain, while the other three were laminated outside on the skin, and they were responsible for powering the device for that was performing the recording of the neural activity, and then a third electrode that was responsible for the transmission of the data outside. So, to better understand, this is the overall structure of the device that we can see here on the left. So, even in this case, the overall device is just this highly flexible shank device where on one side we had the recording sites, while on the other side we have these three flexible gold contact that were laminated on the skull of the animal. Then the skin was sutured, and then with the other three electrodes outside on the skin, we were able to power and get the data from the implanted device. And as we can see from this plot on the right, we're actually able to record the neural activity with an amplitude that was fairly similar to the one of a traditional neuroelectronic device, so based on silicon amplifiers that was wired, while again in our case we had this highly flexible and flexible transistor-based wireless system. More importantly, we were able to perform these recordings in chronic settings in freely moving animals. So just to give a comparison of what we were able to develop compared to what is out there as traditionally implanted device, we see that in the case of a traditional implanted device, we have several rigid components. We have the batteries, we have the wires, we have these silicon-based amplifiers, while in the case of our device, the only thing that we had was, again, highly biocompatible and flexible. So I hope that so far, with all this data, I convinced you that indeed it is possible to create fully flexible bioelectronics. However, this was just the first prototype, but anyway, there are the elements that are needed to build overall bioelectronic devices. And thanks to this work, I was named as one of the 35 innovators under 35 worldwide by the MIT technology review. So now what we are trying to build with my lab, as I mentioned earlier, is a multimodal bioelectronic device. So I've shown so far that we do have a platform that is able to record neural activity, but also heart activity, muscle activity, without the use of any rigid component. Now what we want to do is also adding stimulation techniques to the implanted device. Specifically, we are developing a flexible electrofluidic interface for epilepsy treatment, where basically the flexible device will detect the onset of epileptic activity and then will release a
나 이거를
neurotransmitter,
좀 듣고
GABA,
갈까
that
하면서 들을까?
has shown to
누가?
sorry,
네.
uh, that has shown to block the
음.
epileptic activity. So, describing the overall structure of this device, starting from the top,
누나가 여기 와서
we have
잤다고?
a skin patch that is placed on the skin, and that is responsible for the communication with the implanted device. Then we have the implanted device that has not only the recording electrodes, but also the drug reservoir, where basically after powering from outside, we can deliver the drug to treat the epilepsy. So this is oh, sorry. This is a block diagram of the flexible electrofluidic interface that we are developing. So similarly to the one that I showed earlier, we have this external interface that is responsible to power and transmit the data using the ionic communication. Then we have the implanted device that has the neural recording sites and the drug delivery system. So I've shown how actually indeed it's possible to perform the neural recording using all of these elements. So basically what we're doing now is adding the drug delivery system. And for this, we are using this type of devices that are called ion pump. Even in this case, they're highly flexible and they're able to release drug through electrophoresis. So by applying a voltage between a source electrode and a target electrode. So a source electrode that is in the drug reservoir, and then a target electrode that is in contact with the tissue. And the main characteristics of this type of drug delivery system, of this ion pump, is the fact that they perform this drug delivery that is called dry, meaning that they only deliver the molecule, in our case, the GABA, the neurotransmitter that is needed to stop the epileptic activity, but not the solvents, not the liquid where these molecules are immersed in the reservoir. So that helps with that for not, building up the volume of the brain. So it's something very important if you want to create a drug delivery system that is implantable. And then this overall device with recording electrodes and with drug delivery, again, will be powered with this external interface based on ionic communication. So I hope that with this presentation, I have convinced you that overall, creating fully flexible devices without the use of any rigid components, more specifically conformable multimodal device, so for drug delivery and also recording is not only sci-fi, but it's very much possible. So with that, I want to conclude my talk and of course thank all of my students and my group for helping me with the research, but most importantly, I want to thank each of you for your attention. So if you have any questions, I'm very happy to answer. Thank you.